Welcome to my lecture series, which has a strong focus on genetics. We know that genetics has come a long way, and we can draw a parallel between genetics and other contemporary modern disciplinary sciences. For example, genomics, proteomics, medical and clinical genomics, and personalized medicine, beside molecular and biological sciences. In this quest, however, we have forgotten the fundamental genetics, and fresh input through molecular biology and biochemistry has been ignored. So therefore, this lecture is going to give a holistic view of entire genetics. This discourse will have the molecular insight, therefore, of foundation genetics, and on one side, and on another side we will try to establish a relevance and connection of advanced genetics with modern era disciplinary sciences like genomics, bioinformatics, proteomics, and precision medicine, etc. Coming to the series 1, which consists of laws of genetics, classical and molecular interpretation, and relevance. This lecture is very long, so therefore each of these presentations which are shown here, we will try to present it into many lecture forms. The concept outline of today's lecture is pangenesis and blending hypothesis. We will describe these hypotheses in brief, and preliminary which Mendel concept used. We will describe like genetic material, selection of pea plant, types of fertilization, and of course the terminology and symbols. The old concept was put forwarded by many philosophers, and they concentrated on observing the semen and tried to contemplate how the characters are transferred. One theory, the two philosophers of note are Hippocrates, one of them for example. He gave the pangenesis theory or pangenesis hypothesis between 460 to 370 BC, and opined that genes in the form of pangenes at that time, or particles from various parts of the body, travel to sperms or eggs, through sperms and eggs, and passed on to the next generation. Also, he said that changes which occur during lifetime in the body are passed on to the next generation through sperms and egg. However, we know now that it is not the particle but potential of the body and body features which are inherited, and somatic changes which occur cannot pass to the reproductive cells. The blending hypothesis was put forward in the 19th century by Aristotle, and he observed that offspring inherit traits from parents, and the genetic material is passed on to the next generation by male and female gametes, and then these traits are mixed together in the offspring. So it was something which he explained by saying that blue color, if mixed with yellow color, you have the new color, that is green color. However, with our observations on few examples, like for example red and white flower, or black and brown Labrador coat color, we find that although the red color in the previous case or the black color in the second case, they are dominant one, but the white color and brown color, they reappear. That means no blending take place. Our conclusion therefore that both of these hypotheses, pangenesis or blending theory, are inconsistent with the actual observation of inheritance. Then after that, the early scientists and physicians, they also thought about the inheritance, especially the two Dutch scientists like Leeuwenhoek and De Graaf. They discovered individually the structure of the sperm and the structure of the egg, and these discoveries were much helpful for the inheritance concept. Then after that, the two physicians, one is the French biologist, Pierre de Maupery, who gave the polydactyly, who studied polydactyly, the inheritance and the mutation, and albinism, the lack of pigment in the skin and eyes. And another scientist, British one, J. Adams, who studied different other human traits, and putting together the structure of ovum and studying the human traits, they had some idea that how they have inherited. However, it was left to the Mendel, who discovered the principles of genetics, on the basis of which we could now explain them more clearly and critically. Mendel concepts were first a little bit of his history, that he studied mathematics and science in a monastery school and organized breeding experiments on pea plant, and he presented his result in 1865, and these results were published in Natural Science Association, Brunn, now called Borno, and they were rediscovered about 20 years later by three scientists like Testermack, Correns and Ivory, and they found that the same discoveries were made earlier by Mendel, so they gave credit to Mendel and they came to be known as the principles of Mendel. The success of genetic experiments depend upon the genetic material, selection of genetic material, and we usually concentrate on those organisms which should have short life cycle, large number of offspring, and then economically viable, and maintenance should be easy. They must also have the detectable variation in the form of characters. It should be quite visible and should have separate male and female gametes, that means sexually reproducing organisms, and the crossing experiment should be easier. The Mendel selection of pea plant met the same criteria, rather much more, and not only they were small size, generation times about 4-5 weeks, large progeny, and genome size was also equally small, 7 into 10 raised to power 7 nucleotide pairs, and contrasting forms were quite easily seen in that, and therefore the experiment was followed in this organism. Not only the Mendel chose the pea plants, but also the later geneticists, they successfully used the other genetic material like Drosophila melanogaster, albino rats, that is Rattus norvegicus, mus musculus, guinea, guinea pigs, and E. coli and Caenorhabditis elegans, etc. The floral structure of the pea plant in which the Mendel performed his experiments is quite easy. It is boat shaped, and it has of course the sepal and the petals, the bright red in color, and has the male parts, that is androecium, and gynoecium, that is the female part. The androecium has the anthers or the pollen grains, and gynoecium or the female part has the stigma on which the pollen grains falls, and the self fertilization take place so that you can have the seeds which are produced inside. So self fertilization, I have already explained, it was very useful to have the true breeding varieties of flower color, pod shaped, pod color, seed color, and position of the were obtained as contrasting forms. So these are the 7 characters which Mendel used: height of the plant, position of the flower, color of the pod, shape of seed, and color of the seed. Contrasting form in individual case was quite clear, and by self fertilization clear lines of these were established. Also, cross fertilization, one of the main point which Mendel used after fertilization obtaining the clear. The cross fertilization was achieved by emasculation technique, where you can remove the anthers and pollen grains by cutting them with a scissor, and then to ensure that it should not be contaminated with the outside pollen grains or anthers, you can put a plastic covering around it, and by ensuring that one, you can have the anthers from another plant with different criteria. For example, the anthers from the white plant, white flower color, you can dust it mechanically on the anthers of on the stigma of the violet color plant. So in that way you obtain the seeds, and you can plant the seeds to have the varieties and do the observations. The genetic terminology and gene symbol was another criteria which successfully used by Mendel. And gene symbols which Mendel used was of the 26 English alphabets, and he used uppercase letter and lowercase letter. For example, tall plant with capital T and dwarf plant with the lowercase letter small t. Similarly, he followed capital Y and small y, capital N small r, capital N small a. It was followed by Drosophila geneticist, who used all the 26 letters because there were more genes, and they started using the combination of 2 letter and 3 letter, for example VG and other combination. For example, to avoid the confusion, international genetic symbols guidelines were given, where they say that we should give preference to the mutant gene and mutant character by small w, and the normal one or normal gene should be symbolized with plus sign or plus w or w plus, whatever may be we can use. And another advancement was the use of slash. So here in this case, plus slash plus means homozygous dominant 1, or plus w slash plus w, or w plus slash w plus for denoting the homozygous dominant, homozygous recessive and heterozygous. Similarly, Vg plus and Vg plus for the normal dominant gene, and Vg slash Vg for the mutant. And then as I said earlier, that Mendel was very well versed with mathematics and has the scientific temperament. He also has the analytical mind and discovered 2 methods to analyze the generations. One was the Punnett square method. Of course, it was discovered by another scientist, where the female gametes like capital B and small b, in this case heterozygous, and male gametes again heterozygous, in this ordinate they were put together, and intersection of this as well as male gametes will give you the genotype, and similarly interaction of this with this one will give you the heterozygous, intersection of this and this gamete will give you the heterozygous, and this will give you the homozygous recessive. All these have been shown here: homozygous dominant, heterozygous 2, and homozygous recessive here. Then this was the Vogt method, which was actually used by Mendel, and we can use the F1, that means heterozygous, capital and small r, capital and small r. These are the gametes, these are the male gametes, and then this combination with this one, homozygous dominant, keep this one, use the other one, heterozygous, use the now this time with capital R you will have the heterozygous, with this one you will have small r, small r. So in that way you have the genotype 3:1 and 1:2:1 for the genetic makeup. Thank you very much. We will stop this video only after summarizing the findings of today's lecture. That we started with the old concept which was given by pangenesis and blending hypothesis, and we met with certain criticism. We described that one, and then the physicians they came up with the inheritance of certain human traits like polydactyly, like albinos and other, and they established the link with that of the sperm and eggs with this inheritance. And then we followed the Mendelian concept, and we described about the preliminaries of the Mendelian, like what are the terminologies, what are the gene symbol which Mendel used, and what is the favorable attributes should be for genetic material, especially that of the, and then certain we discuss that one. After this we will go to the next lecture, and that is lecture 2, and there we will describe the genetics of dominance, variation of dominance, law of segregation, and law of independent assortment. Thank you very much.